Hello, I'm Art Modazenik, and I'm with the American Association of Pharmaceutical Scientists and a member of their Committee on Educational Resources. One of our goals is to develop a series of videotapes in which we invite eminent pharmaceutical scientists to speak about some of the experiences that they had in developing research contributions that led to the evolution of pharmaceutical technology and pharmaceutics in particular. One of the special opportunities we had recently was to go to Iowa City, to the University of Iowa College of Pharmacy, and visit with Dean Dale Wurster. Dr. Wurster is a pioneer in pharmaceutics. He's an eminent inventor, educator, and scientist, and we had a special opportunity to ask him about his early work in the development of pharmaceutics. Dale, I'm pleased to have this opportunity to interview you on this series. I know that you were one of the prominent faculty in the 1940s, 50s, and 60s at the University of Wisconsin. The Wisconsin School of Influence in the development of pharmaceutics is very well known, and I would be interested very much in asking you what it was that brought together that particular group of people at that time. How did you emerge at that time as a working faculty in introducing pharmaceutics into the curriculum and into the research programs?
The University of Wisconsin, from the day of its inception, has always been viewed as a strong research institution. In fact, as you know, the PhD degree in pharmacy was given there at a very early time. Thus, there was always a strong scientific approach to the problems of pharmacy. As the dosage form became more important in the delivery system of drugs, it became necessary to design this system on sound scientific principles. With that, the introduction of physical chemistry into the curriculum came about. Since the University of Wisconsin was a pioneer in this curriculum change, it attracted people from other institutions and, of course, students as well. This led the way at that early date. The wisdom of their approach to the curriculum is probably brought out by the fact that it was copied by other great institutions.
During your work and research at Wisconsin, you early on recognized the importance of the release characteristics of the drug from a dosage form, and in fact were an early pioneer in studies doing dissolution on drugs. What led you to these early investigations? What kinds of interests generated your foresight in recognizing that this was going to be an important performance factor in dosage form release?
At that point in time in pharmaceutical research, the disintegration rate of tablets was studied very thoroughly, and many people used different diluents in order to change the disintegration rate. In fact, sometimes this led to tablets that did not disintegrate very well. But in most cases, the tablet dissolved in a matter of a few seconds to a minute. However, the rate-limiting step in the absorption of the drug has nothing to do, for the most part, with the disintegration rate. Therefore, the drug must be in solution in order to be absorbed by the biological membrane or to transport across the biological membrane. Thus, the dissolution of the primary drug particle became more important to me than the disintegration rate, and so I started to study it. I studied it by attacking one physical parameter at a time to see how that influenced the rate.
You early on recognized the importance of biology as a critical discipline that made very valuable use of pharmaceutics. Some of the early work that you had done with percutaneous absorption, the transport of drug across the skin, was very important. What led you to these areas of investigation, and what kind of early leads did you have?
I was led into the biological area by my major professor. He recognized that you must have a good biological background as well as a physical and chemical background in order to do research in pharmacy. Therefore, my graduate work included graduate courses in pharmacology, bacteriology, biochemistry, and others. With this kind of background, it was only logical that I would use it in an attempt to study the absorption of drugs. However, there was one compelling force that was present through all of this. I couldn't imagine that we would be concerned with the physical-chemical system as a delivery system for drugs and stop there. Certainly we all realize that that physical-chemical system is then put in a biological system. It was for that reason that I've devoted emphasis to the biological system as well.
And who was that major professor that helped track you into that area?
Well, he is certainly a great and well-known educator, Dr. Arthur H. Uhl, who was the dean of the School of Pharmacy in later years.
One of the major interests that distinguished your contribution in pharmaceutics from that of many others was that you were also very interested in process technology, Dale. As a matter of fact, your contributions to the development of air suspension coating and fluidized bed theory, which is popularly now known as the Wurster process, is one of the patents that's most frequently cited as the most significant area of pharmaceutical technology in post-World War II. What got you to become interested in particle technology, fluidized bed theory, film coating, and all those areas?
Throughout my entire career, I have always been close to the pharmaceutical industry. And indeed, I had many friends in the industry. Of course, many of our PhD students went to the industry as well, so it was logical to work with them. In so doing, you hear some of their problems. And very often it was suggested to me that I work on the practical problems of coating pharmaceutical tablets. Thus, I looked into different methods to approach the problem, and having known something about fluidized beds and their use in other industries, it seemed like a reasonable place for me to start. Particularly, I wanted to start someplace that the industry did not use at that point in time. I found that the system was equally adaptable not only for tablet coating and making granulations for compressed tablets, but also for microencapsulation and controlling drug release.
Did you actually build any apparatus early on to demonstrate the feasibility of some of these processes?
Oh yes, I built many pieces of apparatus, some of them very crude. In fact, one of them I often include in my talk to show professors that you don't need some real fancy equipment to start something. However, we both know that crude pieces of equipment don't always work that well, so we rapidly progressed to well-designed, industrially produced pieces of equipment.
It's been reported that the Wurster process was one of the best examples of a technology that represented an intellectual property that an organization such as the Wisconsin Alumni Research Foundation was able to promote for the benefit of the university. Was that a surprise to the university and perhaps even to you, the enormous success that technology had in the ensuing years?
Well, certainly when we started it, we could not predict how successful the process would be. However, we did observe that there was an immediate interest in the technology. And while some manufacturers were slow to throw out their present equipment and buy new, nevertheless, they did study the process. And so that set the stage, I believe, for later years. And in the end, it did become one of the best earning patents of the Wisconsin Alumni Research Foundation.
It certainly has turned out to be the benchmark by which a lot of other successes have been measured, and I congratulate you on that accomplishment. One of the ways in which we have attempted to chart the progress in the evolution of pharmaceutics is by looking at the body of literature in terms of reports. And a chapter that you wrote on the compatibility of organic compounds features prominently as a common reference as a major contribution to the early evolution of that field. I don't know if when you penned that chapter you thought it would have such an effect. What were some of the thoughts that you had when you wrote that chapter, and what was the kind of thinking that went into the writing of that?
I started writing the chapter on incompatibilities of organic compounds very early in my career. You may recall there was a series of texts known as American Pharmacy produced at that particular time. And so I was asked to write this particular chapter. I approached the problem with two thoughts in mind. As you know, the pharmaceutical industry had already taken over the production of most products that were prescribed on prescriptions. Nevertheless, about 5 to 10% of the prescriptions still had a list of ingredients that the physician would use in the prescription. Obviously, there could be problems when you put these different chemicals together. So I wanted to use it as a teaching tool to solve that particular problem for the prescription. However, since the products that would also appear on the prescription as already fabricated products by the industry were being produced, I wanted to teach the student also the problems associated in the formulation of products when you put different organic chemicals together. So you could view this text as a solution of prescription incompatibilities and an introduction to formulation work on pharmaceutical products.
In some of your work in percutaneous absorption, Dale, I recall that you and some of your students actually did research with some primitive cells, which pretty much were a precedent for the commercial transdermal devices we see very prominent in pharmaceutical technology today. How did you go about designing some of these early cells, and what was some of your experiences at that time in terms of predicting transdermal drug delivery?
Well, I got interested, of course, in the percutaneous absorption problem because in those early days we had a project to work on protective ointment bases. However, if you're working on protective ointment bases which will inhibit the transport of a chemical across the skin, you better first study the mechanism whereby that chemical can penetrate the skin. Therefore, I started the percutaneous absorption studies. Since this is a diffusional process, there are many physical parameters that have to be controlled. in the simplest sense. For example, the area of the skin that's exposed to the penetrant. With this in mind, I designed a cell in which we could change the conditions so that we could study the individual parameters that were influencing the absorption of the drug. True, these same parameters that we used in those crude cells are built into the devices that are used for percutaneous absorption today.
One of the major roles that you played was as a teacher. You motivated a considerable number of students, and especially in the graduate area. What, in your opinion, Dale, does it take to be a great researcher?
Well, this is a question, Art, that I really do like to answer. As professors, and you are one, you know that we ask our students to be critically analytical when they look at any investigational problem. And in the process of doing this, we all think about certain theoretical aspects which will prevent the project from being a success. We also think of the theoretical aspects which will make it a success. In order to do or carry out research on a particular project, then, I think one must look at it from both of these viewpoints. However, I always told my students, be critically analytical, but come to me with all the theoretical reasons why the project will be successful. I'm not too interested in the reasons why it won't. In the course of my investigational work, several projects that were thought to be very difficult did become successful with this philosophical approach to research.
You've had a very long and productive career in various areas of pharmaceutical sciences as an educator, a researcher, a teacher, and even applying things to pharmaceutical technology in the industry, Dale. What was it actually, though, that motivated you to seek a career in pharmaceutical sciences, to seek an undergraduate degree in pharmacy and a graduate program in pharmacy?
My major professor, Dr. Rule, is mainly responsible for my interest in graduate work and obtaining the PhD degree. When I was an undergraduate student, he called me in on several occasions, but I remember very well the first occasion where he explained to me what graduate work was, what would be expected of me, and what the career opportunities would be. Other professors taught graduate work with us, and these are well-known names to all of you, Dr. Lloyd Parks and Dr. Louis Bussey among them.
During the 1950s, there were a series of teachers' conferences that were held that charted the course of the new curriculum for colleges of pharmacy at that time, as you will recall. Some of those conferences were held in Madison, Wisconsin, and played a major role in revising the curriculum, emphasizing physical chemistry and pharmaceutics. You were an important contributor to those discussions, Dale. Would you comment on how those discussions came about and what some of the arguments were at that time?
Yes, the teachers' conference was held on the University of Wisconsin campus. It was the first conference of its type and was carried on for many years, as you know. At this conference, we had the usual program of lectures in the various scientific areas. And at this point in time, of course, physical chemistry had already been introduced on the Wisconsin campus in the College of Pharmacy curriculum. With this, we discussed the introduction into the extended curriculum, which would result from the pharmaceutical survey, which had been initiated, I believe, in 1947. There was considerable discussion as to whether courses in physical chemistry per se, which we then chose to call physical pharmacy, should be taught, or whether the physical chemistry should be integrated with the pharmacy courses that we already taught. There was considerable difference of opinion. The Wisconsin idea held that we would give specific courses in the area where some other institutions favored integration.
At one time at Wisconsin, you were the leading contributor to the evolution of the continuing education program, and one of the most prominent aspects of that program was the industrial conferences held at Land O'Lakes, Wisconsin. How did those come about? Who were the principal contributors and the people who helped bring those conferences into being?
Well, the faculty had been interested in doing something that would bring us closer to the pharmaceutical industry. We, of course, had many projects with them. However, we wanted to serve them as well as work on projects. So, particularly in this regard, Professor Louis Bussey and Takaru Higuchi were very, very important in introducing the industrial seminar. I played some role in this and participated in the planning and also in giving lectures and devising ways to help finance it.
Can you recall any of the industrial participants in those series of programs who might have had a major role behind the scenes in introducing industrial support for those conferences?
Well, you know, I'm afraid that I will leave somebody out, but I will take a chance and mention a few. As you know, Walt Enns at Upjohn was very interested in the new pharmaceutics, if you please, and he was a strong supporter. Larry Wheeler at Park Davis and Company was very helpful. Tom Masick at Merck and Charlie Jarosky at Pfizer. All of these people helped us so much that I think we could not have put on these seminars without them.
Dale, the work that you and your students did in publishing so much on your dissolution research became very apparent to the industry as a principal way in which the quality of a product could be measured. In fact, corporation after corporation began to do dissolution testing rather than disintegration testing to measure the release of a product. This ultimately had important implications in bioavailability. Did you anticipate that it would have such a remarkable application?
Yes. In fact, we were concerned at all times that the blood levels would be influenced. And to make the drug available to the body was certainly the primary objective. There are many people who followed up and utilized the dissolution technique to help them predict some of their absorption rates and other parameters in pharmacokinetics. One name that cannot be left out is that of Dr. Eno Nelson. Dr. Nelson is certainly the pioneer of that particular type of work. I might add parenthetically that many nights when we were both working. He was a graduate student and I was a young professor. We had long talks about the influence of dissolution on some of these other parameters.
Dean Arthur Uhl from the University of Wisconsin School of Pharmacy had the special talent of attracting scientists from outside of pharmacy to the faculty at the university. And this made a major influence in the development of the curriculum. How was he able to do that, and what factors did that have in the graduate program in particular?
Well, Dean Ewell always said, find bright young people and get out of their way. And with this philosophy, he found a bright young physical chemist by the name of Takeru Higuchi. I wish very much that Takeru could be sitting here with us this afternoon and answering your questions. However, as everybody in pharmacy knew, he made a great impact on pharmaceutics and the teaching of pharmacy and the research in pharmacy. We will ever be indebted to him for that. But he was trained initially as a physical chemist. He was unusually adept at being able to apply physical chemistry to our pharmacy problems. And I guess I need not say any more about this because he is known throughout this country and internationally for those contributions. As a quick aside, I might say to you, however, that we were longtime colleagues. He and I started on the University of Wisconsin faculty the same day.